So good morning, everyone, once again. Today I will be presenting some data on a peculiar small non-coding RNA called miR-218 as it pertains to the preclinical model of depression. And to take a step back at first, psychiatric disorders are characterized by an early onset, as has been mentioned by many speakers before me. They tend to emerge in adolescence, and coincidentally, this is the same time when the prefrontal cortex, a brain region responsible for high-order cognition, is undergoing organizational alterations, which no doubt have a functional implication. And for example, to take the major depressive disorder, which, as Dr. Paus has mentioned, is one of the most prevalent psychiatric disorders, we observe a disproportionate impact where females are at least twice as likely to experience the symptoms as males. Now, in relation to this conference, there are significantly fewer number of studies that are looking at biological underpinnings or biomarkers for psychiatric disorders when it comes to looking at the period of adolescence. And actually, the same is true for early life. So clearly, there's this big gap in studies that I think is the reason why our work is so important. Now, in our laboratory, we find that DCC, which is a guidance cue receptor for Netrin-1, guides neurons in the mesocortical limbic pathway to innervate the prefrontal cortex during adolescence. And we find that microRNA called miR-218 is a post-transcriptional regulator of DCC. So it binds DCC, thus inhibiting its expression, clearly suggesting that both have a role to play in the prefrontal cortex during adolescence. Now, in postmortem human studies, we find that in individuals with depression who died by suicides, DCC is significantly upregulated, whereas miR-218, its post-transcriptional regulator, is downregulated, hence suggesting that there is this clear molecular dysregulation in patients with major depressive disorder. And of note, microRNAs are particularly interesting as they are small and they are very stable when it comes to collecting the tissue or preserving it over long periods of time. And they are so small that they can be engulfed by these small vesicles called exosomes, which are able to pass the brain-blood barrier and achieve this long distance communication via the microRNA signaling. And this, of course, has a significant strength as we may infer or use the microRNA levels in blood as proxy of what may be going on in the brain, as of course is a challenge when it comes to looking at human cohorts. Now, the present study aims. Depression emerges in adolescence, as I have mentioned, and is twice as common in females as in males. So here I assessed miR-218 expression over development in blood and the prefrontal cortex, and identify how altering its levels in adolescence may impact behaviors in adulthood. Now, briefly to mention the methods, I used C57 black mice at different developmental periods. So early adolescence, mid-adolescence, and adulthood. I collected brain tissue and blood, which then was used to assess the miR-218 expression levels using the RT-qPCR. And to the point of my last aim, as I mentioned, I used antisense oligonucleotides that directly bind miR-218 to degrade it. And this is administered via intranasal spray to target the nervous system systemically. And this takes place in adolescence, which then our mice are allowed to grow into adulthood to undergo several behavioral paradigms, but here in particularly, I'll be talking about the elevated plus maze, which is a measure of anxiolytic and risk-taking like behaviors. Now onto the results. On the left side, I will be showing some data found in male mice, and this data has already been published. And on the right, it will be in females, some of our recent findings. And what I'm showing here is that in the prefrontal cortex, we observe that miR-218 is gradually increasing over the lifespan in male mice, whereas in females, we see this advanced what appears to be like maturation in miR-218 by mid-adolescence already. Now in blood, we see that in males, there is this mirroring of what is going on in the prefrontal cortex, as I have shown, the gradual increase in miR-218 levels. However, intriguingly, in female mice, this is not true, and in fact, it's the complete opposite. miR-218 levels are being reduced over the lifespan. All this to say that there is this clear postnatal development trajectory. miR-218 expression in blood and brain seems to be sex-dependent. Now, as I mentioned, the findings in males have already been published as part of this study. And before I proceed with the next result, I would like to mention that these studies have all been on male mice, as the preclinical model for depression, the chronic social defeat stress, is reliably reproduced in male mice. Thus, from now on, I will be showing studies in males. And so just to mention that in this study, we collected blood in adolescent mice, and in adulthood, these mice underwent the chronic social defeat stress, which is where the experimental mouse is experiencing chronic stress over several days by this larger, more aggressive CD1 mouse. And at the end of the stress, we used a social interaction test to segregate the mice into two groups, either susceptible or resilient to the social interaction defeat. And so susceptible mice would spend more time in the corners, whereas resilient would spend more time with the aggressor as compared to the baseline time traveled. And what we observe is that in adolescence, mice that had upregulated levels of miR-218 were actually the ones that were more likely to be susceptible to the behavioral abnormalities after the chronic defeat. Whereas if they had lower miR-218 levels, they were more likely to become resilient. And so in the next slide, I will be showing how downregulating miR-218 would impact the behavior in adulthood. So, as I mentioned, I used the antisense oligonucleotides, or antagomir, to downregulate miR-218 in male mice in adolescence. And in adulthood, I measured their behavior in the elevated plasmase, and I observed that mice that received antagomir, or had downregulated levels of miR-218, in adolescence show more time spent in the open arms of the elevated plasmase as compared to the control, thus suggesting that indeed miR-218 has this role in, at least in the anxiety-like and depressive-like behaviors. And with that, I would also like to briefly mention that we are also currently working in collaboration with several other labs on samples that have been also non-invasively collected, so either blood or saliva, from a cohort of adolescents, some of whom are experiencing depressive symptoms, to then profile global microRNA levels and be able to differentiate the expression of microRNAs, some of which may be indicative of risk or resilience of psychiatric vulnerability in these adolescents and as they grow into adulthood. So these are longitudinal cohorts. And to me, this means a critical implication, as these studies will be able to help, of course, in future prevention and intervention, as well as longitudinal tracking of the disease. So with that, to briefly summarize everything I've spoken about today, psychiatric disorders emerge in adolescence as the brain undergoes maturation. miR-218 is a modulator of DCC expression in the prefrontal cortex, both of which are altered in the prefrontal cortex of individuals with depression. The developmental trajectory of miR-218 is sexually dimorphic, as I have shown in blood and the brain. And targeting miR-218 in adolescents has lasting effects on behavior, potentially having a therapeutic application, yet it is critical to keep in mind that this effect on behavior may differ between males and females and remains to be investigated in future studies as well. And with that, I would like to thank you for your attention. Here's a recent picture of the beautiful Florist Lab, and I will take any questions if there are any. Thank you.
Thanks, Alice. Great presentation. So interesting. So any questions? Maybe while people are thinking, one question I have a couple, but one is really related to this particular microRNA and the mechanism by which it may be inducing changes that you described. You of course talk about DCC, but that must be one of hundreds of genes that are targeted by this microRNA. So do you know, have you done any say gene ontology analysis on the genes that are targeted by this particular microRNA? What else might be influenced?
Yeah, that's an excellent question. And you're absolutely right that microRNAs may bind several gene targets at a time. And for example, we looked at other gene, also guidance cue receptor called ROBO1 in the prefrontal cortex. So for example, as I was talking about the postmortem studies, when we, so postmortem studies were also very much replicated in our animal models. And so what we find is that miR-218 is impacting DCC, whereas it does not impact its other gene target, which is the ROBO1, also a guidance cue receptor. And so in this particular example, so you can see there is an effect, a specific effect, but in terms of gene ontology, you are right that this is something that important to look at, and I would expect it would have many implications for neurogenesis as well as synaptic regulation. So, I think I would look at it specifically when I am doing the human genetic studies and looking at several targets at the same time.
Okay, thanks.
I had a question, Alice. I was wondering if when you look at the microRNA in the blood and the brain tissue, if you did anything like correlating within the individuals to see like how similar it was there.
Yeah, so you mean how it relates when I'm looking at miR-218 in blood and brain within the same mouse?
Right. Because you have samples from both, like the same mouse, both brain tissue and blood, right? So I would, I guess, like expect them to be highly correlated, but then the data from the female in particular suggests that maybe you'd see the opposite.
You're absolutely right. I didn't show this data. It's a correlation, and in males, it is a positive correlation, where in blood and brain, there are, again, very much a positive correlation, but in females, very much negative correlation. So I find it to be particularly interesting, and if I may be so bold, that, we see a huge prevalence in psychiatric disorder, or particularly depression, in females. And so perhaps this may be one of the reasons, one of the molecular underpinnings where that underlies this difference in the prevalence.
That's really unexpected and fascinating. It's
Yeah.
so good that you did the work in females, or else I might not have known.
Yes, exactly. So as with all studies, it is critical to be able to assess both males and females for us to have a better understanding of the disease as well as the differences between the sexes.
Other questions? Well, let me ask more then. Just following up on Lani's question. So you're saying that across the different animals coming from different developmental stages, at that population level, you see positive correlation between the brain and blood in males and negative correlation in females.
Yes.
Now that kind of begs the question, what regulates the regulator, right? So microRNA is a regulator of gene expression. What regulates microRNAs? And obviously, what would be then different along those lines between males and females?
So to me, microRNAs are epigenetic, not markers, players, and I would expect that environment very much has an impact on microRNAs, which in turn are then regulating their target genes. So that would be my answer, very much experience dependent.
Well, but do we know anything about, let's think for a minute about the periphery, right? The systemic environment. And obviously the obvious difference between males and females, at least in terms of what's circulating in their blood, are hormones, sex hormones, right? So do we know anything about how sex hormones influence levels of this particular microRNA in blood?
This is an excellent question. Fortunately, I'm not able to speak about it at this moment, but there are studies in our lab that have been specifically looking at males and females as well as the hormones. So you're absolutely right that there we would expect there to be an association as well. I just can't speak of a specific example right now.